As you're looking at pathogens, you have to, there's going to be a difference in the way viruses spread, their symptoms, and how they respond to treatment versus bacteria. And they don't even, in this case study, they don't even consider the other kinds of infections. All right. In fact, let me just change that. There's other kinds of infections that they don't really consider, but I'm going to put off to the side. Fungi. All right. And protist infections. Not to mention parasites. So, could someone close the door? And whoever closes it is going to have to open it again for whoever shows up late. Protists and fungi, protists, as well as parasites. These all can cause disease and have similar qualities as far as how they spread through contact, fecal oral, which means somebody doesn't wash their hands and they touch food or doorknob and then you touch the doorknob, that kind of thing. They focus on the difference between these two because of the idea of vaccinations versus antibiotics, right? And we talked about this. Everybody should know this. If you don't know this, you should get ready because you're going to get a quiz on this week, right? Antibiotics are for bacteria. Yeah. Antibiotics are for bacteria, and inoculations or vaccines are for viruses. Yes, we have some antivirals. They don't work as well. They're nothing like our stores of antibiotics. And with each there are issues. With antibiotics, what you're going to find is that there are resistant strains. As you're going along in the curriculum, you're going to find that there are resistant strains. There are strains that the antibiotics no longer work. That confuses you as a doctor, you as a nurse, you as a public health official are going to find it difficult when you give someone an antibiotic and it doesn't work. You might be thinking, well, it could be viral. But it could also be that this infection, this bacteria is resistant to the antibiotic. So that happens. And by the way, I'm going to mention this again. I had mentioned it previously. The way they become resistant is called evolution. They change the frequency of alleles in a population. That's called evolution. And just as an aside, I'm just going to keep bringing out this tree, right? Do you understand how this works? This flow chart, right? These antibiotics, there are resistant strains of bacteria. Do you know what a strain is? So you have E. coli as a species. You have E. coli, let's call it E. coli A or E. coli B, right? They're both E. coli, but B is resistant and A is not. Does that make sense? So the same species of bacteria, but the one group and we call the groups of bacteria a strain. Does that make sense? Okay. So the reason that they become resistant is because of evolution. That being said, I want to make a note that we will be doing a lab on this antibiotic resistance. So as soon as the equipment comes in, we will be doing this lab. All right, and you'll come across it. It's in your curriculum. So I'm just going to put anti-resistance. Does that make sense? Everybody see where I'm going with this? Okay. So this is really, when you are reading about antibiotic resistance, remember you will have a lab that will make it clearer. Okay? Yes? No? Maybe? We're all good? All right. So that's your really big deal, is how do you tell the difference between a viral infection and bacterial infection? I believe as you're going through the cases, you're seeing that there's a lot of people in a lot of different situations, and you have to do kind of an investigation on who touched who sneezed where, who was hanging out with who, correct? And you have a whole list of people showing up to the clinic with various symptoms, and you have to figure out which ones do they all have the same disease? Is it just spreading? Does the disease show up with different symptoms in different people? You know, these are research points, right? These are questions you have to research. So there are questions you have to go and do research on. Do not expect that all the answers are going to be in front of you. Does that make sense to you? And some of the questions are, how do I tell the difference? Between, bacterial and viral. Infections. How do I know it's not? How do I know it's not it's not some kind of toxin? Did they get food poisoning? Is this food poisoning? I don't know. Like, how do you tell? So those are questions you should be researching. And we'll get into more of this later, but right now, one of the tests that some of you have already done, right, that you've come across, according to what you're telling me, is two big tests, DNA sequencing and then the other one is another test you've come across is something called PCR.
I just got PCR.
Excellent. These are two tests that we usually run. They usually run on viral infections. You can run them on bacterial infections as well. But the viral infections are going to be where you're going to focus. All right. So you're going to focus these tests on viral infections because, interestingly, with bacterial infections, what we can do is we can take a sputum sample. We can take your saliva, we can take your nose drippings, or in the case of a wound, we can take the sample from your wound. We can put them on something you're going to get very familiar with as soon as we start doing labs, is these plates called Petri dishes.
Oh, I've seen those before.
Okay, we have a whole ton of them. And we're going to lay inside the Petri dish, we're going to make this thing called agar. And agar is a gelatin. I can't get this color right. So it'll have different colors depending on what we're looking at. Usually it's kind of like a tannish color.
We're gonna go for two.
That's okay. It doesn't have to be perfect. Just wanted to be less crazy. Oh, and it didn't take. Why? I don't know. All right, so whatever. So it's why is it yellow, man? But I'm choosing it. Let me choose it. All right, whatever. It'll be yellow for now. It doesn't matter. So, anyways, the agar is going to be like a jello gelatin. It will not be yellow, and you'll pour it in. What we do is we pour it in to these. We melt it in a microwave, and or boil as we make it. We cook it. We put it in. It's almost like cooking jello gelatin. Have you ever made jello gelatin? Yes, you've eaten jello.
You can just say Jello.
All right. Yeah, it's a brand. So, anyways, the jello or the gelatin. This is a gelatin. It's made of seaweed. It's made of seaweed carbohydrates that form a matrix. So it acts like a partly solid. It's full of made with water, and you can add anything you want to. You can add all kinds of foods, and you can add antibiotics. So, for instance, you can add, food, and you can add antibiotic A. So what you can do is you can grow. This is a side view of the. This is a side view of the agar plate. I'm going to look at it down from the top, and when I look at it from the top, it's going to be round. And you're going to do this in class in the labs. So try to pay attention, and we'll go over it again. But I just want to make sure you're paying attention. And you have food in there, and you have an antibiotic. You have one that has the antibiotic and another one that doesn't have the antibiotic. So you have your control group. And you take the snot, you dilute it so that it's nice, so you can spread it easily. Or the wound juice, the pus or the saliva or whatever you're testing. And you spread it on the, let me try to make this little. You spread it on the agar plate like that. So you have that snot in there. Now you leave it, you put it in an incubator, you wait. Wherever there was a bacteria, it will start to multiply because there's food. So let's look at the control group. The control group is no antibiotic. So you wait a day, maybe two, and just a few hours really. And what you'll see is you'll start to see wherever there was one single bacteria, you'll see a whole colony now. These are colonies. These colonies of bacteria are called plaques. Plaques. You've heard of plaque on your teeth? Those are bacterial colonies. All right? So they're growing. There's billions of little bacteria in there. The one with the antibiotic is not either going to have less or no growth, no bacterial growth. If there's no bacterial growth, then you know that the antibiotic is working to kill that bacteria. But you still don't know what that bacteria is. So we have all these chemical tests. First of all, you can look at bacteria under a microscope, and you can identify, is it a rod? Is it a coci? Does it have a flagella? right? Do they group in clusters? There's all kinds of characteristics that help you identify what kind of bacteria you're dealing with. And there's all kinds of tests you can keep running. Does it turn purple when I add this chemical? Does it grow does it need this to grow? Will it eat blood? Does it live without oxygen? Does it need does it die if there is oxygen? So there's all kinds of tests that you can run, and that tells you what kind of bacteria you have. Does that make sense? So if you have there's all kinds of little tests you can do. Now they're much faster, but you still have to grow them. So we can do that. And in fact, when you first go in the hospital room and you're sick, if they're worried about something like this, it's obviously more than one person just coming down with a cold or something, they'll do these growths. They'll send your snot and your blood to the lab, and this is what the lab does. They grow the bacteria to see what kind of bacteria you have. And if it's the disease remember, there's bacteria all over you all the time, correct? So now there's pathogenic. What's pathogenic mean? Versus, forgot the H. Versus what? Pathogenic versus, what does pathogenic mean? Wow, nobody looked that up, huh? Again, you got to look this up. Yeah, so what's a pathogen? You're right. There's a pathogenic means there's a pathogen somehow involved. It's okay. It's really easy. Just try to remember it. Put it on an index card or something. It means disease causing. It's really easy. So it's disease causing versus non-disease causing. So there are some bacteria, and the one that a lot of people know, a staph. You've heard of staph infections, right? Staphylococcus aureus, flesh-eating disease. Staph infections are very famous, so that's why I always use it as an example. Would be under disease causing. So it's pathogenic. Not all staphylococcus are disease causing, but Staphylococcus aureus is one good example. Why do you have a computer open? Anyone. I'm not just picking it. Well, everyone does. I told you. That's right, I forgot. That's my bad. I told you I'm old. Leave me alone. Stop staring at me and judging me. All right, so anyway, Staphylococcus, disease causing, we're all good. So that's how we do it if it's a bacteria. In a viral infection, though, you can't do that. You can't look at a virus under a microscope, at least not easily. We can look at an electron microscope, which we have one on loan in Mr. Krishnan's room. We have an electron microscope, a scanning one. I would love to take you after school if anybody wants to and go look at some and actually use it to look at things very closely. It's kind of cool. The only problem with a scanning electron microscope is you can't look at living biological tissue. Or you can't look at anything that's alive. It can't be fresh. So that's one issue. At least not with this one. So, but with a virus, you need an electron microscope to look at the virus. They're that small. In fact, they're called viral particles. They're not. You can't look at a microscope and say that's what this is. And they don't grow like bacteria. You can't put them on a plate. They don't grow on their own. They need to actually be an infection. The only way they make more copies of themselves is if they're actively infecting a cell, because the cell is what makes copies. Is that clear? That's why, as I told you earlier in a previous lesson, that's why most scientists don't consider them alive, because they can't make copies of themselves. Is that fair? Yes? Okay. So now that brings us to and I guess we're going to have to finish this tomorrow. That brings us to PCR and DNA sequencing. And before I can talk about that, I have to talk about DNA a little bit. And we have to talk about how we copy DNA. And those of you that are doing honors biology, have you started doing DNA yet? No. Okay. And really, before I get to that, so tomorrow, I would actually like to talk about proteins, because the first lab that you're going to have to do, assuming we get the lab equipment sometime in the next century. Sorry, getting a little frustrated. Proteins, we really should talk about things like antibodies, because that's really the first test we do is antibodies. Who's seen the test for COVID? What do they do? COVID's a virus, right? Everybody knows that? Yeah. They stick your nose, they get the snot, they put in a little tube.
And then you set it in this small thing and wait for it to change color or not, and if it changes.
Right. Very good. That's right. If you do the home test, it works pretty much the same way. And most of the home tests, for sure, most home tests, and a lot of tests that you go to the clinic to do, they use antibodies. We could also use a more expensive test called RNA. Both have to do with the production of protein. So perhaps we should start with proteins first and then move on to DNA. And I believe in honors biology, you're working on proteins. Is that correct?
Yeah.
All right. So, but we will extend your knowledge on proteins, and then we'll talk about how DNA can be tested, because you're going to have to. That's what PCR and DNA sequencing is. But I think it's probably better for us that you do this lab that the AP kids have already are in the middle of doing. And I think you need to, obviously you as non-AP kids are not going to go into as much depth as they are going into it. But I'll present it to you. You guys can go into it as much depth as you want. But there's just some basic ideas that you need to know about proteins to really be able to understand how we can test PCR and sequence and how that impacts what kind of pathogen we have in this case. Does that make sense to you? All right. I know it seems like a lot, but I'll be walking you through this material, all right? I want you to do research. I want you to keep doing the case, get this stuff in your head, know that you've heard of PCR. Just to give you some vocabulary words to really start working on, I would start putting in those index cards. PCR stands for polymerase. Chain reaction. Now, you need to know that ACE means that suffix means enzyme. Again, we'll have to talk about what that is. And the other thing is, you'll have to know what a polymer is. Right? And so those are kind of two big, and most enzymes, I'm going to go ahead and put this as most enzymes are proteins. I have to tell you, that's absolutely true. However, some, not same, some, again, it looks like same, weird. Some enzymes are RNA. So you see, in both cases, for understanding PCR, for understanding DNA replication, to understand sequencing, you really have to understand what an enzyme is, you have to understand what a protein is, and why it matters why DNA matters. All right. Those of you who came in late, please come here.
I don't know what's wrong with you. I said I don't know what you do.
Polymer means a large molecule made of repeating units. And we call those units monomers. It sounds complicated, doesn't it? Well, let's break this down a little bit. Poly here means many. Does everybody know that? All right, so poly means many. Mer means unit. So many units. Okay? A monomer is one unit. A polymer is many units. So, still a little complicated, right? Okay, well, who here has not played with Lego blocks? So everybody's played with Lego blocks. Is that correct? Mm-hmm. Oh, well, then you understand this. There's nothing else to it. So when you have a Lego block, you have a unit, correct? Is that right? And when you have this unit in a Lego block, what does this unit represent in this definition? That's a horrible Lego. What is it? If I look at this definition, and I'm telling you that if you played with Legos, you understand this. What is the single Lego represent? One unit, which is what? A monomer, correct. So this is your monomer. And then if you take a bunch of these, and stack them up on top of each other, what do we get?
A polymer.
The whole thing is a polymer. No problem. Easy peasy, lemon squeezy. A bunch together, a polymer, a single one is a monomer. Now, where does it get tough? It gets tough because they're not Lego blocks, are they? No. Let's take a look at this. And this is you're going to have a lab tomorrow. This will be the lab, the AP kit, not tomorrow, Thursday. Actually, no, because Thursday I'm taking the chess team, either Thursday or Friday, depending on when the chess team is having their competition at the Guardians. So I'm taking the chess team to the Guardians again to play the baseball team in chess. Tomorrow's Wednesday. You have parent-teacher conferences. Are we clear? Mm-hmm. Okay. So these are not Lego blocks. As simple as this idea is, biological systems use different monomers. They don't use, but you have different Lego blocks, don't you? You have round one, not round. Do they have round ones?
Yeah.
Yeah, kind of round ones. They have rectangular ones, square ones, long ones, short ones. Well, there are different monomers for biological molecules as well. Let's break it down to the four main biological molecules. The first type is going to be carbohydrates. The second, and let's say protein. This is stuff you know from health. This is stuff you know from food, right? From eating, correct? Nucleic acids. That's one that might be new to you. Maybe not. Probably not. Hopefully not. And the last kind is lipids. But you know them as fat, right? Fats, oils, etc. Monomer and then polymer. Lipids do not have monomers. They do not form polymers. Do not form polymers. It's a little more complicated. Fatty acids, I guess you can consider them a type of monomer, but they never form polymers. All right, so anyways, you don't have to worry about lipids. There's only 3 that you have to worry about: monomer and polymer. Obviously, we're worried about these 2 in this class, although really in honors biology, you're going to have to know all of them. And for those of you that had honors biology, you should already know them. All right. So a monomer for a carbohydrate is known as a monosaccharide. That's a mono means one, saccharide means sugar, one sugar.
Can you zoom in a little bit?
Monosaccharide. Again, my messy handwriting. I wish I didn't have to write it. So maybe if I typed it up ahead of time, but then I would go crazy. Saccharide. Is that clear? Mm-hmm. All right. So monosaccharide is a sugar. So you would know this. You know what this is. There's glucose. You've heard of glucose, correct? Yes or no?
Yes.
Yes. It looks like this. It's 6-sided, has a hexagon. Usually they'll draw it as a hexagon. You've heard of another monosaccharide called fructose, correct? High fructose corn syrup.
Mm-hmm.
So that is by the way, there's an oxygen there, but whatever. That is a 5-sided sugar, 6 carbons still, but 5-sided. There's another monomer that you're going to need to know eventually, and most of you that I had know it already. And there's this thing called ribose. It's actually a 5-sided sugar that has 5 sides. All right. Oh, that doesn't have this doesn't have All right. So those are the big ones. There's others. There's several others like galactose and other sugars as well. All right, what's a polymer? There's 2 big ones you've heard of or you will hear about. There's cellulose and there's glycogen. And there's others like chitin. Chitin is the sugar that makes the exoskeletons of bugs. When you crush bugs, what you're crushing is a sugar shell.
Is that a C after the Y?
Yes.
Okay.
So cellulose, you know it as paper or wood. That's what plants make their structure from. Oh, there's another one that you probably should know. Is starch. Please note that you've come across these words in your life, have you not?
Yeah.
Starch, you've heard of starch, for sure. Chitin, probably not, but that's what the sugar that the exoskeleton of a bug is made of. Cellulose is just paper, wood. That's what plants make their structure from. Are we all good with that? I'm going to come over here and do proteins while you're writing. Proteins, the monomer for proteins, you know what they are. And you're going to be working with them on Friday. They're amino, what? Acids. I'm going to draw a crazy structure. You should get to know them, especially if you're in honors bio now. It's actually not that hard. But the monomer looks like this. There's a central carbon. There's a hydrogen. This is a central carbon. Like to put an asterisk on it. There's this variable group. It's called an R group. And you're going to need this to do the lab. That's why I'm drawing this now. You need this for Friday. There's that's bonded to C. Every carbon. Now this is so important, guys. 9th graders or 10th graders that are taking honors biology. Let me be very clear. Carbon always has 4 bonds. Period. If it does not have 4 bonds, something is wrong. Now, I say that, but obviously there's intermediate forms that may or may not have 4 bonds. But if you see a drawing on a test, multiple choice question, or if you draw something that has a carbon that has more than 4 bonds or less than 4 bonds, something's wrong. Is that clear? That's an easy way to help yourself. The other thing you should know is oxygen always has 2. Unless it has a negative charge on it. Then it could have only 1. Is that clear? Mm-hmm. All right. So see, what if I double bond this carbon to an oxygen? How many more bonds does that carbon need? The bonds are represented by the lines. Each line is sharing 2 electrons. I'm not going to go into it. Each line is sharing 2 electrons. So there you have it. How many more bonds does carbon need? How many bonds does carbon make? 4. So let's count them. 1, 2, 3. It needs 1. Do you see that? 4. Do you see it? Yes. Because it's important that you see it. So that is bonded to another oxygen, which needs 2 bonds, which ends up being bonded to hydrogen, and this is called an acid group. This is an acid group because it releases this hydrogen here, is easily released, and this is a hydroxide. The whole thing is known as that hydroxide. Let me change this because that could be very confusing. So let me just say this. An OH group is also known as a hydroxide. Now, these two together, this C double bond O, OH, all this together is known as a carboxyl group. Or a carboxylic acid. Carboxylic acid. So this is the acid part of the amino acid. This big C double bond OH. On the other side, you have nitrogen. You have a 2 hydrogens. This is known, this group over here, NH2, is known as an amino group. So there you have a carboxyl group or carboxylic acid, an amino group. Now do you see where you get amino acid? Amino acid. There's 20 of these different ones. You're going to learn all of them on Friday. Each one has its own properties, but this is your monomer. So you can take a bunch of these and put them together, and what do you make? If I take this carbon with C double bond O, H, double bond O, OH, that's a carboxylic acid group.
We can't see it.
Here's the hydrogen. Oh, is it too small? Is that better?
Yeah.
There's your hydrogen. It's bonded to this R group. This R group means it's a variable. There's 20 different ones. I have them here for you in color. They're drawn out in color. Little symbols. All right? You're going to make a protein. On Friday, you're going to start on Friday. You'll continue to play with them for a couple days. Friday, Monday, and Tuesday. It's so important that you understand this. We're going to play with it for 3 days. But before we do, you need to know what you're looking at. Does that make sense? So you have that end, and on this end, you have your amino group, NH2. So that's now you got another, you're going to put them together. How do we put these together? Well, you need 2 of them. And again, there's 20 different R groups. So how many amino acids do you have in your body? Usually, there's more, but how many? If there's 20 different R groups. How many different amino acids do you have? 20. You can have R that's hydrophobic, R that's hydrophilic. Did we cover hydrophilic and hydrophobic? Have you covered it in Honors Bio yet? You all know what it is, but they haven't covered it yet, so I'll have to go over that tomorrow.
It's almost time to go.
It's almost time to go, but let me show you something. When you build out these monomers, you put them together, unlike Lego blocks, you have to actually remove something because you can't connect them without removing something. Do you see that?
Oh, don't you?
Because carbon can only make 4 bonds. Nitrogen can only make 2 in this situation, or 3 in this situation. You have to remove something so you can connect these 2. So what turns out leaving is the OH from this amino acid and an H from this amino acid to make,
O2H.
that's right, H2O. We'll finish this, we'll continue this tomorrow.